Good afternoon, and welcome to AP Biology Semester 2. We're in Big Idea 2, Free Energy, and today we're in Chapter 7, Membrane Structure and Function. And we're going to be in Section 7.1, talking about the cellular membranes. Cellular membranes are fluid mosaics of lipids and protein. All right, let's start with life at the edge. The plasma membrane is the edge of life, the boundary that separates the living cell from its surroundings. A remarkable film only about 8 nanometers thick, it would take over 8,000 to equal the thickness of this page. The plasma membrane controls traffic into and out of the cell it surrounds. Like all the biological membranes, the plasma membrane exhibits selective permeability. That is, it allows some substances to cross it more easily than others. One of the earliest episodes in the evolution of life may have been the formation of a membrane that enclosed a solution different from the surrounding solution while permitting the uptake of nutrients and elimination of waste products. The ability of the cell is to discriminate in its chemical exchanges with its environment and is fundamental to life. And it is the plasma membrane and its component molecules that make this selectivity possible. In this chapter, you will learn how cellular membranes control the passage of substances. The elegant structure of a eukaryotic plasma membrane shows the protein that plays a crucial role in nerve cell signaling. This protein restores the ability of the nerve cell to fire again by providing a channel for a stream of potassium ions to exit the cell at a precise moment after nerve stimulation. The green ball in the center represents one potassium moving through the channel. In this case, the plasma membrane and its proteins not only act as an outer boundary, but also enable the cell to carry out its functions. The same applies to the many varieties of internal membranes that partition the eukaryotic cell. The molecular makeup of each membrane allows compartmentalized specialization in cells. To understand how membranes work, we'll begin by examining their architecture. Okay, so lipids and proteins are the stable ingredients of membranes, although carbohydrates are also important. The most abundant lipids in most membranes are phospholipids. The ability of phospholipids is to form membranes. To form membranes is inherent in their molecular structure. A phospholipid is an amphipathic molecule, meaning it has both a hydrophilic region and a hydrophobic region. Other types of membrane lipids are also amphipathic. Furthermore, most of the proteins within membranes have both hydrophobic and hydrophilic regions. How are phospholipids and proteins arranged in the membranes of cells? I'm so glad you asked. You encountered the currently accepted model for the arrangement of these molecules in Chapter 6. Woo. I need some water. In this fluid mosaic model, the membrane is a fluid structure with a mosaic of various proteins embedded in or attached to a bilayer of phospholipids. Scientists propose models as hypotheses, ways of organizing and explaining existing information. We'll discuss the fluid mosaic model in detail, starting with the story of how it was developed. Okay, so we're going to the membrane models. And this right here where we're going has to do with scientific inquiry. Yay! Scientists began building molecular models of the membrane decades before membranes were first seen with the electron microscope in the 1950s. In 1915, membranes isolated from red blood cells were chemically analyzed and found to compose of lipids and proteins. 10 years later, two Dutch scientists, E. Gorter and F. Grindel, reasoned that cell membranes must be phospholipid bilayers. Such a double layer of molecules could exist as a stable boundary between two aqueous compartments because the molecular arrangement shelters the hydrophobic tails of the phospholipids from water while exposing the hydrophilic heads to water. Okay. In 1972, S. J. Singer and G. Nicholson proposed that membrane proteins are dispersed, individually inserted into the phospholipid bilayer with their hydrophilic regions protruding. This molecular arrangement would maximize contact of hydrophilic regions of proteins and phospholipids with water in the cytosol and extracellular fluid, while providing their hydrophobic parts with a non-aqueous environment. In this fluid mosaic model, the membrane is a mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. And because models are hypotheses, replacing one model of membrane structure with another does not imply that the original model was worthless. The acceptance of or rejection of a model depends on how well it fits observations and explains experimental results. A good model also makes predictions that shape future research. Models inspire experiments, and few models survive these tests without modification. New findings may make a model obsolete. Even then, it may not be totally scrapped, but revised to incorporate the new observations. The fluid mosaic model is continually being refined. For example, recent research suggests that membranes may be more mosaic than fluid. Often, multiple proteins semi-permanently associate in specialized patches where they carry out common functions. Also, the membrane may be much more packed with proteins than imagined in the classic fluid mosaic model. Let's now take a closer look at membrane structure. Okay, we're going to go to the fluidity of membranes. Membranes are not static sheets of molecules locked rigidly in place. A membrane is held together primarily by hydrophobic interactions, which are much weaker than covalent bonds. Most of the lipids and some of the proteins can shift about laterally, that is, in the plane of the membrane, like partygoers elbowing their way through a crowded room. It is quite rare, however, for a molecule to flip-flop transversely across the membrane, switching from one phospholipid layer to the other. To do so, the hydrophobic part of the molecule must cross the hydrophobic core of the membrane. The lateral movement of phospholipids within the membrane is rapid. Adjacent phospholipids switch positions about 10 to the 7 times per second, which means that a phospholipid can travel about 12 micrometers, the length of many bacterial cells, in 1 second. Proteins are much larger than lipids and move more slowly, but some membrane proteins do drift, as we will find out later. And some membrane proteins seem to move in a highly directed manner, perhaps driven along cytoskeletal fibers by motor proteins connected to the membrane proteins' cytoplasmic regions. However, many other membrane proteins seem to be held virtually immobile by their attachment to the cytoskeleton. A membrane remains fluid as temperature decreases until finally the phospholipids settle into a closely packed arrangement and the membrane solidifies. Much as bacon grease forms lard when it cools. The temperature at which a membrane solidifies depends on the types of lipids it's made of. The membrane remains fluid to a lower temperature if it is rich in phospholipids with unsaturated hydrocarbon tails. Because of kinks in the tails where double bonds are located, unsaturated hydrocarbon tails cannot pack together as closely as saturated hydrocarbon tails, and this makes the membrane more fluid. The steroid cholesterol, which is wedged between phospholipid molecules in the plasma membranes of animal cells, has different effects on membrane fluidity at different temperatures. At relatively higher temperatures, at 37 degrees Celsius, the body temperature of humans, for example, cholesterol makes the membrane less fluid by restraining phospholipid movement. However, because cholesterol also hinders the close packaging of phospholipids, it lowers the temperature required for the membrane to solidify. Thus, cholesterol can be thought of as a temperature buffer for the membrane, resisting changes in membrane fluidity that can be caused by changes in temperature. Membranes must be fluid to work properly. They are usually about as fluid as solid oil. When a membrane solidifies, its permeability changes and enzymatic proteins in the membrane may become inactive. For example, if their activity requires them to be able to move laterally in the membrane, the lipid composition of cell membranes can change as an adjustment to changing temperature. For instance, in many plants that tolerate extreme cold, such as winter wheat, the percentage of unsaturated phospholipids increases in autumn, an adaptation that keeps the membranes from solidifying during winter. Okay, so now we are going to talk about the membrane proteins and their functions. Now we come to the mosaic aspect of the fluid mosaic model. A membrane as a collage of different proteins embedded in the fluid matrix of the lipid bilayer. More than 50 kinds of proteins have been found so far in the plasma membrane of red blood cells, for example. Phospholipids form the main fabric of the membrane, but proteins determine most of the membrane's functions. Different types of cells contain different sets of membrane proteins, and the various membranes within a cell each have a unique collection of proteins. There are two major populations of membrane proteins: integral proteins and peripheral proteins. Integral proteins penetrate the hydrophobic core of the lipid bilayer. Many are transmembrane proteins, which span the membrane. Other integral proteins extend only partway into the hydrophobic core. The hydrophobic regions of an integral protein consist of one or more stretches of nonpolar amino acids, usually coiled into helixes. The hydrophilic parts of the molecule are exposed to the aqueous solutions on either side of the membrane. Some proteins also have a hydrophilic channel through their center that allows passage of hydrophilic substances. Peripheral proteins are not embedded in the lipid bilayer at all. They are appendages loosely bound to the surface of the membrane, often to expose parts of integral proteins. On the cytoplasm side of the plasma membrane, some membrane proteins are held in place by attachment to the cytoskeleton. And on the cytoskeleton side, certain membrane proteins are attached to fibers of the extracellular matrix. These attachments combine to give animal cells a stronger framework than the plasma membrane alone could provide. Okay, now, one of the functions of the protein is to transport. So a protein that spans the membrane may provide a hydrophilic channel across the membrane that is selective for a particular solute. Other transport proteins shuttle a substance from one side of the other by changing shape. Some of these proteins hydrolyze ATP as an energy source to actively pump substances across the membrane. Now, proteins are also used for enzymatic activity. A protein built into the membrane may be an enzyme with its active site exposed to substances in the adjacent solution. In some cases, several enzymes in a membrane are organized as a team that carries out sequential steps of a metabolic pathway. Proteins are also used for signal transduction. A membrane protein or receptor may have a binding site with a specific shape that fits the shape of a chemical messenger, such as a hormone. The external messenger signaling molecule may cause a shape change in the protein that relays the message to the inside of the cell, usually by binding to a cytoplasmic protein. Proteins also are cell-to-cell recognition. Some glycoproteins serve as identification tags that are specifically recognized by membrane proteins of other cells. They are also used as intracellular joining. Membrane proteins of adjacent cells may hook together in various kinds of junctions, such as gap junctions or tight junctions. They also attach to the cytoskeleton and extracellular matrix. Microfilaments or other elements of the cytoskeleton may be non-covalently bound to membrane proteins, a function that helps maintain cell shape and stabilizes the location of certain membrane proteins. Proteins that can bind to ECM or the extracellular matrix molecules can coordinate extracellular and intracellular changes. Okay, so I need to, okay, that's it for that one. And we're almost done with this section. So now we're going to talk about the role of membrane carbohydrates in cell-to-cell recognition. Cell-to-cell recognition is a cell's ability to distinguish one type of neighboring cell from another. It is crucial to the functioning of an organism. It is important, for example, in the sorting of cells into tissues and organs in an animal embryo. It is also the basis for the rejection of foreign cells, including those of transplanted organs, by the immune system, an important line of defense in vertebrate animals. Cells recognize other cells by binding to surface molecules, often to carbohydrates on the plasma membrane. Membrane carbohydrates are usually short, branch chains of fewer than 15 sugar units. Some are covalently bonded to lipids, forming molecules called glycolipids. Recall that glyco refers to the presence of carbohydrate. However, most are covalently bonded to proteins, which are thereby glycoproteins. The carbohydrates on the extracellular side of the plasma membrane vary from species to species, among individuals of the same species, and even from the cell type to another in a single individual. The diversity of the molecules and their location on the cell surface enable membrane carbohydrates to function as markers that distinguish one cell from another. For example, The four human blood types designated A, B, AB, and O reflect variation in the carbohydrates on the surface of red blood cells. Alrighty, we're getting ready to wrap this up, and we're just going to talk a little bit about synthesis and sidedness of membranes. Membranes have distinct inside and outside faces. The two lipid layers may differ in specific lipid composition, and each protein has directional orientation in the membrane. When a vesicle fuses with the plasma membrane, the outside layer of the vesicle becomes continuous with the cytoplasmic inner layer of the plasma membrane. Therefore, molecules that start out on the inside face of the ER end up on the outside face of the plasma membrane. Okay, so that's about it. I'm going to stop here for now. And the next time we meet, we will be talking about membrane structure results in selective permeability. Until next time, ta-ta.